Today, our group will be focusing on nuclear magnetic resonance spectroscopy, where we'll be talking about the uses of 13C NMR spectroscopy. Firstly, let's look at the brief overview of what is 13C NMR spectroscopy. 13C NMR spectroscopy is the application of nuclear magnetic resonance spectroscopy to carbon. Although carbon-12 is the most abundant isotope, it has zero net spin and will not be detected by NMR. Hence, carbon-13 is used. Moreover, carbon-13 NMR spectroscopy is a high-resolution method to study chemistry and biochemistry.
To talk about the use of carbon-13 NMR spectroscopy to study the brain metabolism. By detecting resonance from carbon-13, which is the only stable isotope of carbon having a magnetic moment, carbon-13 NMR can be used to follow the fate of carbon-13 labeled substrates in the brain, both in viral and virtual environment. Viral means a living organism and virtual means a laboratory environment. And here are the three stages of NMR spectroscopy to study the brain metabolism. The first stage is the infusion of carbon-13 labeled substrate. Next is the detection of carbon-13 labeled metabolites following substrate consumption. And the third one is the metabolic modeling of measured carbon-13 enrichments to quantitatively derive metabolic flux. In general, these three stages are closely interconnected, and each of the stages imposes constraints on the other two. Hence, all the three must be designed depending on the specific metabolic pathway that are to be investigated. The advantage of carbon-13 NMR is that it's probably the most elaborated, chemically specific tool to follow the metabolic fate of carbon-13 labeled substrates in the brain, both in viral and virtual. Thus, it measures metabolic process when they occur in the intracellular environment. And it provides unique information which is not accessible from previously used approaches. As we mentioned earlier, carbon-13 NMR spectroscopy detects resonance from carbon-13, which is the only stable isotope of carbon with a magnetic moment. However, this causes carbon-13 NMR spectroscopy to be a relatively insensitive method as compared to other conventional methods, because of the low natural abundance of carbon-13 among all other isotopes of carbon. However, this insensitivity can be improved noticeably by using carbon-13 enriched substrates.
Next, I'll be sharing about examining the structural chemistry of natural organic matter using 13C nuclear magnetic resonance spectroscopy. 13C nuclear magnetic resonance spectroscopy is a useful tool for examining the structural chemistry of natural organic matter, such as investigating wood and its properties. For example, we can study wood tissues and investigate its molecular dynamics. A more in-depth study of wood can be seen through the study of coarse woody debris. Solid-state 13C NMR spectroscopy was applied to coarse woody debris in different stages of decomposition. The debris were collected from many different types of forest climates, for example, a subtropical, a cool temperate, and a subalpine forest. The purpose was to test 13C NMR spectroscopy's applicability to characterize organic chemical composition of debris of broad-leaved and coniferous trees from different climatic conditions in more detail. With more detail, we can understand the decay dynamics of not only leaves but also leaf litter and soil organic matter. Moreover, 13C NMR spectroscopy is generally compatible with those of approximate analysis and has a potential to provide useful indicators of long-term decomposition of woody debris. Over the years, 13C NMR spectroscopy has become an important method in establishing the chemical composition of organic soil matter. Some pros of 13C NMR for the study of coarse woody debris include avoiding problems that most techniques use. For most techniques, determining the chemical nature of natural organic matter in soil, sediment, and water require prior extraction or concentration steps that are not significant and create artifacts. However, 13C NMR spectroscopy does not require all those steps. Moreover, 13C NMR analysis is the preferred method to determine the relative proportions of carbon functional groups, such as alkyl, aryl, and carboxyl groups. Despite its advantages, 13C NMR spectroscopy has some disadvantages. Firstly, it gives little information at the scale of molecules, where it could cause scientists to utilize whatever spectrometer is available to them. Secondly, certain elements, such as the presence of iron, could cause some interference, where it could cause the detection limit for carbon to be higher than intended, giving inaccurate results. The presence of different elements could be avoided by increasing the time in which the experiment is conducted over. However, the effect cannot be fully removed, causing certain inaccuracies. Lastly, certain peaks in the spectroscopy can be ambiguous, as the carbon can be in different positions in the molecule but have similar resonances.
Now, I will be talking about 13C NMR for wastewater treatment. I would first give a short overview about the wastewater treatment process before explaining the applications of NMR in this process and its pros and cons. As sewage is a complex mixture of sand, organic and inorganic components, it is processed in treatment plants in three main steps. The primary step, which uses screens to remove large debris such as sand and gravel, followed by the secondary step, which involves the biodegradation of organic matter, where the sewage is mixed with a controlled population of bacteria known as activated sludge in digestion tanks. Finally, the tertiary step, which uses filtration methods like reverse osmosis and disinfection procedures such as the use of chlorine and UV light to further purify and sterilize the reclaimed water for use. Now moving on to the applications of 13C NMR. I will discuss three applications of 13C NMR in wastewater treatment. 13C NMR is useful in ensuring that the effluent, also known as reclaimed water, is safe and within safety guidelines by identifying illegal sources of contamination. It is also able to detect malfunctioning wastewater treatment plants by studying the different components such as complex organic matter in the effluent, sludge, and wastewater. It is also able to study the functionings of wastewater treatment process for improvements. The 13C NMR spectra is often divided into 5 main chemical shift regions, representing alkyl carbon bonded to heteroatoms, aromatic carbons, carboxyl carbons, and carbonyl carbons. By using NMR and observing the resultant spectra, it is possible to identify compounds in the effluent and ensure that the composition of the effluent complies with safety guidelines for safe consumption, as several residual and complex molecules may still remain after the treatment. Next would be the use of NMR in the detection of malfunctioning wastewater treatment plants. Organic matter degrades to give humic and fulvic acids. Humic acids are composed of alkyl aromatic units such as carboxylic acid and ketones, while fulvic acids are more aliphatic. Therefore, the extent of the decrease of polysaccharide content and the increase in production of structures with more aromatic and aliphatic polyesters through the formation of alkyl linkage observed from the NMR spectra will be able to inform us on the efficiency of the wastewater treatment plant in treating organic matter. 13C NMR can also be used to gain a better understanding of the treatment process and therefore enable us to make improvements. For example, it is used to study the metabolic processes of bacteria in the activated sludge. 13C labeled propionate is broken down by bacteria through products like valerate. This is studied to understand how organic matter is degraded through different cycles to figure out ways to improve efficiency. This is important for the safe recycling and disposal of organic waste after the effluent is collected. Now I will share the advantages and disadvantages of using 13C NMR. The advantages are that it is versatile in characterizing both overall chemical composition as well as individual species in a wide range of mixtures. It also requires little pre-treatment, making it highly suitable for environmental studies. Also, it is rapid, non-destructive, and can be coupled with other techniques such as high-pressure liquid chromatography to improve detection limit. However, NMR experiments can require a long time to finish if not homogenized. It must be free of particles because the presence of solids deforms the homogeneity of the local magnetic field, causing a broadening of signals.
In recent years, 13C NMR spectroscopy has found a use in cancer diagnosis. Researchers generally recognize abnormal cell metabolism as a sign of cancer, and thus aim to study cell metabolism of glucose in order to determine whether a patient has the condition. Cancer cells preferentially carry out anaerobic respiration to produce energy, in which glucose is converted to pyruvate and then to lactate. In this procedure, the substrate, which is either pyruvate or glucose with the first carbon atom labeled as carbon-13, is injected into the subject's bloodstream. Some studies have documented hyperpolarization of the substrate before it is injected, which helps amplify the resultant signal 10,000 times. The tissue or organ of interest is studied to detect large concentrations of 13C labeled lactate, which demonstrates a higher signal-to-noise ratio in tumor cells than in healthy cells. This method allows for obtaining of both images and spectroscopic data, and can potentially be used to monitor cancer cells' response to treatment. The main advantage of this method is that it is non-invasive. It allows for the study of tissues and cells without running the risk of damaging the sample. 13C NMR spectroscopy is also safer for researchers and subjects alike as compared to traditional imaging methods. The former does not feature radiation as it makes use of carbon-13, which is a stable isotope. The latter, however, tends to use radioisotopes, which give rise to ionizing radiation that can cause cell damage and even cancer. The absence of such radiation in 13C NMR spectroscopy also helps researchers to save money. Disposal of radioactive samples can prove costly, and 13C NMR spectroscopy eliminates the need for such a procedure. However, this method is not without its drawbacks. It is quite insensitive. The strength of the signal generated is highly dependent on the concentration of 13C nuclei present in the sample. This can be overcome by strengthening the magnetic field or by using a hyperpolarized substrate as mentioned earlier. 13C NMR spectroscopy also involves working in an environment with a strong magnetic field. While there are no known risks to human health from doing so, this can adversely affect the functioning of electronic equipment, such as computers, that are used in the study. Objects made of magnetic material may also become attracted to the magnet and thus pose a safety hazard. Fortunately, electronic equipment designed to function in a high magnetic field is becoming increasingly available, together with surgical instruments made of non-magnetic material. Another way of overcoming the problems associated with working in a strong magnetic field is to place a shield around the magnet to reduce the fringe fields. Finally, 13C NMR spectroscopy is very sensitive to movement. Even seemingly small movements such as the beating of a heart can distort the signal. In studying organs such as the brain, a technique called cardiac gating must be applied, in which the obtaining of images is timed with the subject's cardiac cycle. This is necessary due to the movement arising from the pulsating manner of the blood flow.
Next, I will be presenting the use of 13C NMR spectrum in food science. NMR technology was initially used in the late 1940s to elucidate the structure of molecules in organic chemistry. However, the diverse applications of NMR spectroscopy in food science were delayed until the 1980s. With the development of NMR instrumentation and improved programs to collect and analyze the data, NMR's applicability has recently rapidly expanded in the field of food science and technology. A wide range of NMR food-related research has covered various fields of food science, including food microbiology, food chemistry, food engineering, and food packaging. Take oil study as an example of 13C NMR researchers. High-resolution 13C NMR spectra are more complex than 1H spectra. They contain much more structural information, chemical shifts, and intensities. 13C NMR resonances of fats and oils can be grouped into 4 well-defined spectral regions. Carbonyl carbons ranging from 173.3 to 172.8 ppm, unsaturated carbons ranging from 132.0 to 127.1 ppm, glycerol carbons ranging from 69.1 to 61.6 ppm, and aliphatic carbons ranging from 34.3 to 14.0 ppm. Information contained in this spectra has been employed for edible oil authenticity determination and quality controls, including the analysis of fatty acids composition and distribution of fatty acids in the triacyl glycerol molecule, the free fatty acid, iodine value, and diacyl glycerol determination, the analysis of minor components, the oil stability prediction, the determination of polar components, and oil colors. All this data suggests that, with only one analysis, NMR allows the determination of a large number of components with very little or without any manipulation of the oil samples that nowadays need many different analyses. In food science, barriers to development of NMR spectroscopy instruments are primarily due to high cost, the expertise involved, and safety issues related to magnetic field maintenance. Low-field NMR and MRI are relatively more accessible to food researchers due to low cost and easier maintenance, but their applications are still limited. In order to ensure proper data acquisition and analysis, more NMR trained staff are needed in the aspect of food application. Food researchers also face challenge to establish standard operation procedures of NMR/MRI analysis for specific categorized food products due to the complex nature of foods.